Hi. Well, welcome everyone. It's nice to see you here today. Again, I'm Mike Apley. I'm a veterinarian from Kansas State University and a clinical pharmacologist. My background includes private practice in central Kansas. I spent 4 years as a feedlot consultant in the early 90s and have taught at Iowa State University, now at Kansas State University in the veterinary schools. What I teach is food animal medicine. I've done a little bit of stint in the clinics there in the field service. I teach clinical pharmacology and then production medicine courses. So this is my area of interest, is the use of drugs in food animals. And today we're going to talk specifically about cattle. And pipe up with a question anytime. If you'd rather wait till the camera's off, that's fine too. And we'll have plenty of time for questions, comments, thoughts afterwards. So feel free. So this is my commute every day. Every once in a while we do have a traffic jam when cows are being moved or out. My 18-minute commute, or my 18-mile commute through that takes 21 minutes, so a little different than living out here, but enjoy it every day. So next slide, please. Well, cattle go through a variety of production stages, and I imagine we have people in here who grew up on a cattle ranch, and we have people to who this is kind of new, so we'll start out by getting all on the same page. So up in the upper left, that's a pretty shiny big heifer with her mom on some Flint Hills grass. And they go from there to maybe to the upper right, which is a group of steers I owned back in Iowa on our pasture that are in a grass phase. So these are what we'd call stockers. I bought some nice big strapping steers to put out on grass, and we grazed them over the summer and then sold them in the fall. Down in the lower left is some southwest Kansas short grass pasture with cows and calves on them. And then on the right is a feedlot situation up in northern Nebraska. Cattle move all over, and over there on the stockers, a lot of cattle will go through that phase. About 30% of our cattle come into a feedlot for the finishing phase as calves. The rest are older and they've come through something such as that grass phase, or what we call a backgrounding operation, where it's lower stress, lower energy feed, a lot more room, and we kind of ease them into the feeding situation. But that's the way we raise cattle in the U.S., a variety of those, and there may be some other production phases we don't see on there. Okay, next slide, please. So what do we use antimicrobials for? And I use the term antimicrobials because I'm one of those academic guys, and antimicrobials technically includes both the antibiotics and the synthetic compounds. So antibiotics are produced by one organism to inhibit the growth of another, and we modify those. So for example, there are penicillins that the nucleus was produced by one organism and we modify it and make more. And an antimicrobial is a synthetic compound which does the same, and it also includes antibiotics. An example of the synthetic ones are the fluoroquinolones. So I'm sure most of you have heard of ciprofloxacin. That's a fluoroquinolone, which is a totally synthetic compound. So I use antimicrobials. We'll use antibiotics today kind of as the common usage term indicating all of them. So we have several uses: therapy, control of disease, or growth promotion. So we'll talk about therapy first. Obviously, very few of us would argue that that's a needed application of antibiotics. And our goal in the cattle industry is to do as much as we can to make sure we don't need to use those for therapy. It's something we do when we need to, but we try to avoid through management. Control of disease has two phrases that are sometimes confused, and they're not interchangeable. Prophylaxis is when I use an antibiotic ahead of the disease challenge to try to prevent cases. And one example is if you were to go in for a surgery where they felt the need for prophylaxis, they might give you a shot of antibiotics before and then during the surgery to make sure an infection didn't occur, or try to prevent it. Metaphylaxis is a practice in the cattle industry where we have a group of animals that we know are a mix of ones that are just starting to display disease, ones that we suspect are in the incubation period for the infectious disease, and ones that may not yet be in the incubation period. When we apply an antibiotic to the entire group, we term that metaphylaxis. And we'll talk about that a little bit more in the slides to come. So, very good. In our next slide, about 26% of the cattle in feedlots receive a metaphylactic treatment of an antibiotic on arrival, and that means an injectable. We do give it to the whole group, but the animals come through the chute where we process them. They get vaccinations, they get treated for parasites, and then we would give those cattle selected for this practice an injectable antibiotic. And most of those probably the effect of the antibiotic is there maybe out to 2 weeks at the most. A lot of them about 10 days. It wears off, it leaves their system, and then they'll stay there another 6 to 8 months in that facility after that happens. That is from the National Animal Health Monitoring System, or NAHMS Feedlot 2011 study. If you're interested in learning more about today's practices in feedlots, you can just Google that or go to the National Animal Health Monitoring System site and look up the Feedlot 2011 study. And it's a survey about all the practices and the way we do things in feedlots. So metaphylaxis was selected based on these criteria according to the producers that were surveyed, and it includes arrival weight, how far they traveled to get to the feedlot, which is a surrogate for stress. How much stress do we think we have on these? Whether they were preconditioned, which we talked a little bit about before, where they went through a different phase before they came to the feedlot, where they come from, if we know they have a previous antibiotic treatment, if we know they've had previous vaccines, and then the breed type and the general overall quality of the animal. So metaphylaxis is applied to an entire group. In the way here, it's injected, and it's done according to criteria. It isn't just we pick a group or we do it to everyone. We have criteria to decide which ones it's done. So next slide, please. Now the third category, or growth promotion, is used in cattle on feed, and the primary one is the group called the ionophores. These are a polyether antibiotic. They are a true antibiotic. They are produced by one organism, and we use them to alter the microbial environment in the rumen. So according to the NAHMS study in 2011 of feedlot practices, about 90% of feedlot cattle receive this drug in the feed. So we'll talk a little bit later about estimates of how much is used for growth promotion. In cattle, this is 90% of the cattle receive this drug. Now the ionophores, as far as I know as a clinical pharmacologist, have no known or even theorized relation to human health or to drugs used in human medicine. We haven't identified resistance genes that could be included with other resistance genes, and we select for those by using this drug. None of that occurs, to my knowledge, as of today. So they're very safe in that manner, and they do some really nice things for us. They increase the efficiency of production by about 8%. So it takes about 8% less feed to produce the same pound of animal. They also prevent a disease called coccidiosis, which is an intestinal parasite that can induce a lot of stress and cause them to be at higher risk for other illnesses. And in some cases, they can increase rate of gain. There's another thing to understand about the ionophores is that by law, if another drug is not cleared to be used with this, we can't use it. So the fact that 90% of the cattle are getting that tells us that there's only 10% that could possibly be getting another. And from my experience, it'd be pretty rare that they're getting anything then. So this is the growth promotion we really use. Rate of body weight gain, improved efficiency, et cetera. And it does really benefit us on cost of production and the environmental impact, because it cuts our feed by about 8% that it takes to raise that same pound of animal. Next slide, please. Here's what I expect to happen on growth promotion, and we hear a lot of varying things, but I'm talking to people that have these drugs. They own the drugs that we're talking about. And except for the ionophores and the bambamycins are another group that are used also for altering performance. When neither of these are on the medically important list for humans, I expect that we'll see the others gone in 4 to 5 years. As soon as Guidance 213 is approved and finalized and says, here's how we're going to make Guidance 209 happen, we have 3 years. The companies have 3 years to either show that those have an efficacy effect and prove that, and that doesn't mean that they just say, we think it has an efficacy effect and the FDA says it's okay. It means they're going to have to bring evidence of efficacy and then show safety also. But within 3 years, any uses of growth promotion antibiotics that are medically important are to be phased out once Guidance 213 is finalized. Next slide, please. So there's some myths that I hear that I want to address and make sure everyone's aware of some of the facts behind these. And number one is one that I just saw in a newspaper article again today, is that 70, or you may hear 80% of all of the antimicrobials used in the U.S. for human and animal uses are used for non-therapeutic use in food animals. This report is seriously flawed. It's referred to, and one of the reasons is there's an example of a couple of the antibiotics that were calculated in the figures for swine that aren't even marketed in the U.S., so it tells you a little about the research in there. And then I'm the lead author of a group of people that did an estimate that was published in 2012. So if you just go on to PubMed and put in Apley, MD, for my initials, this will be the first one that comes up. It's one of my most recent articles. And we used swine 2006 data to go through and estimate actual swine use, and then validated it with some companies that would share confidentially with us their 2006 sales data, and found out that we were pretty close to what actually was sold. Pop back to that slide, please. There you go, thanks. And what we found was that in swine, and we're going to repeat this work for cattle based off the feedlot study, in swine we found that actually for growth promotion, about 16% of the antibiotics used in 2006 that are medically important to humans were used for growth promotion. All the rest was prevention or control or therapy. And then our study was validated, so drastically different combinations in there. And the other thing about that 70% figure is, remember the ionophores in cattle? About 40% of the antibiotics sold for veterinary use in the U.S. are those ionophores. So that figure's in there also. Next slide, please. Here's another one that we use antibiotics as a replacement for management. We go back to the example of the metaphylaxis, where about 26% of the cattle that arrive into feedlots receive that. Well, we don't want to have to do that. So that's why we have preconditioning programs, we have vaccination programs. For example, also in the NOMS 2011 study, 96% of the cattle arriving at feedlots are vaccinated for respiratory disease. A lot of those that are preconditioned have already been vaccinated for that. They've already been acclimated to the feeding and watering systems, et cetera, as one way to try to prevent that. We do use genetic selection, and that doesn't mean we go and alter them. What that means is we're able to trace back performance in the feedlot, including health performance, back to the farm of origin. And then we can select for the heifers we retain as future breeding animals, and we can select bulls that give us the best health. And we're getting better and better evidence that health is really pretty highly heritable, and that's a valuable tool for us. And also the management of cattle movement. In the U.S., we do move cattle from the southeast to feeding facilities in the middle of the country. And so some of those preconditioning yards are in the middle. We shorten the movement, we let them develop their immune system, respond to vaccines before moving them on. There's a lot of different movement schemes, but we use those to try to avoid using antimicrobials whenever we can. Next slide, please. There's a lot of regulatory control for this use in food animals, and sometimes it's portrayed as being really loose. But there are actually extensive regulations governing both the approval of antimicrobials, which we'll talk about in a minute, and also their use. For example, any use of an antibiotic other than on label requires a veterinarian's involvement. So the veterinarian has to design a regimen and then assign an exaggerated withdrawal period before that animal can be harvested. Any extra label use in the feed is prohibited. So if you want to see what the uses are in beef cattle, you can look at the labels, and if we go outside of that, that's a violation of the law. If you want a site that's really useful for finding out what is approved for certain uses, there's one called Animal Drugs at FDA. If you go to that site, you can search by application, by generic drug name, by trade name, by a variety of different categories, even route of administration. So what's approved for in-feed use for cattle, that site will let you do that. Then you can take what's called the New Animal Drug Application Number, or NADA number. It's a three-digit number, and you can go to another site. So if you Google FDA, CVM, Freedom of Information summaries, you can actually see all the work that was done to approve that drug. You can see the safety work, the toxicity work, the efficacy work, summaries of that. So you can find out a lot in a publicly available information. Next, please. And this is one that really bothers me because I've been involved in some of these processes, and that's that it's really easy, or there's kind of like a lower level of evidence for a drug approved for food animal use than one that's approved for human use or any veterinary use compared to human use. In fact, it's extremely rigorous. It takes many millions of dollars. It's still not quite the cost of a human because we don't have the human trials. But we have extensive toxicity work. We're required to show safety for the animal, safety for the person handling the drug, safety as far as potential toxicity through the food animal products, and for ones approved today, we have to show microbial safety through 2 different types of qualitative risk assessments. So it's really very, very extensive. Just the toxicology testing early on can be several $1 million just to get that going. Next, please. So I've got several things that I want to encourage us all to include in a productive dialogue on resistance. And I'm not here today to tell you that resistance doesn't happen. It does. And I'm not here today to tell you that we've got separate groups of bacteria in humans and animals. That's not true either. But we have a lot to talk about and a lot to look at. And so there's certain things we need to have included in that. And one of the things to talk about is just what do we mean by resistance? And just before I came here today, I spent time at the committee that meets twice a year where we set susceptible and resistance cutoffs for bacteria on the veterinary side. So when you see that an organism is susceptible or resistant and it's veterinary, we're the ones that set that. And we consider multiple kinds of data to set that. And our goal is to get cutoffs where if it takes this much drug to inhibit the bacteria, we think it's going to work clinically at a certain dose administered in a certain way for a disease. If it takes this much, don't use it. We don't think it's going to work. Those are called clinical breakpoints or clinical cutoffs. There's another kind called epidemiological cutoffs. These are designed to look at large data sets of bacteria and to determine when the population has changed. So for example, if we have this distribution today, we might set our cutoff right here, and we start to pick up bacteria out here, we're going to say they're resistant by the epidemiological cutoff. We don't know if that means resistance to being treated in a clinical setting or not, but we're saying, hey, the population's changing. And what happens in our discussions sometimes is we start mixing epidemiological cutoffs and clinical cutoffs, and one of the things I try to do is bring that back to let's get on the same page and talk about what we're really talking about. Our discussion should be about specific antimicrobial pressures that are being used, rather than all antibiotic use good, all antibiotic use bad, all growth promotion good, all growth promotion bad. We need to talk about specific uses, and I think we made a really good example about the ionophores. And when we start to throw those in big chunks, big conversations, it's just one of the antibiotics, we get misled. And the other thing is the bacteria of interest, and I'm going to come back to that in a minute, but it's very important that we don't talk about some bacteria as being resistant and then seamlessly go to food animal uses when any type of correlation would be really, really suspect. And then that interaction between the antimicrobial and the bacteria of interest is very, very important to discuss, specifically about how we're talking about interaction and how it might select for resistance. Okay, next please. And then the outcome of that interaction. There's some very interesting data out there. There's a paper by Lonergan, which I often refer to, that shows what happens to intestinal E. coli resistance, the number of bacteria actually truly resistant in the gut after an injection of a long-acting antibiotic. And what happens is if you look at 3 or 4 or 5 days, you go, wow, it's changed what's happened with that flora in the gut and how it's changed to reflect different susceptibility levels. But if you look back in 2 weeks or 3 weeks, it's back to where it was. So there are changes, and some of them go away pretty quick. And then we also need to talk about transfer, combined resistance elements. It gets pretty complicated, but we need to keep all these things into discussion. Next slide, please. You have to click twice. There you go. So we have some resistance challenges on the veterinary side in our own species of pathogens, but since we're talking about human-animal use interactions today, what are the pathogens that we really need to talk about, and which ones do we talk about in relation to food. Enterococcus faecium is one that's showing a lot of resistance today. In the human side, Enterococcus faecalis and some of the Enterococcus species can be found through the food chain. Staph aureus, we hear a lot about methicillin-resistant Staph aureus. The type predominantly found in food animals in the United States is not the one causing our outbreaks in the United States. It can transfer to humans and be carried to humans, but the work shows that it's basically lost its human virulence factors. And so the one that causes the Staph aureus outbreaks, the hospital MRSA infections, is almost without exception one that's human-derived or circulating in the human population. There are some studies you may have heard about overseas that have shown that they've tracked an MRSA from food animals to people. There's two, I think, in a Danish study. They came from small farms, and those are pretty rare. Klebsiella, Acinetobacter, Pseudomonas, human pathogens, Pseudomonas aeruginosa, very, very rarely. We may see that as a mastitis pathogen, but it's not a widespread pathogen we deal with in food animals at all. Those three, it's pretty hard to argue that food animal production is having any type of effect on those as the Enterobacter. Next slide, please. So those were called the escape pathogens, and after being initially brought out to the public in 2004 and 2009, they did an update, and they're still the primary drivers of concern. Now, I'm not intentionally ignoring foodborne pathogens, Salmonella, Campylobacter, and yeah, we got to talk about those with what we're doing in food animals and what effect it might have on those important foodborne pathogens. The escape, the CDC again in 2009 shows MRSA, vancomycin-resistant enterococci, fluoroquinolone-resistant Pseudomonas are very, very, very important. We're starting to see more and more focus on what's called carbapenem-resistant gram-negative pathogens, and those are starting to come around to us through Europe. We see a few of them here. Those are very concerning because of the fact that the carbapenems are one of our most potent drug groups, very, very important, and to see that coming forward as resistant. Unless it's a linked gene somewhere to something we're doing in food animals, we do not have carbapenems in food animals. In the United States, every once in a while one might be used in a dog or a cat. To use those in a 100-pound Rottweiler would probably cost you $150 a day. So it's not something that we would see as an option in food animals, either from cost or availability, or even if you were to consider it extra label. Right now, more people are dying of MRSA infection in U.S. hospitals than HIV and AIDS and tuberculosis combined. So it is a big, big concern. Next slide, please. So there's a few things I wanted to sum up with before we have a discussion. Is antimicrobials are a very valuable resource for human and animal diseases. And the use of antimicrobials in cattle is both benefits and risks. When you look at the ionophores, I see a lot of benefits. I see very few risks at the current time, and that is a number that's included in the numbers we talk about. I think the important thing is to evaluate both in the decision-making process, and I encourage you in your discussions on this issue that we keep track of those discussion points I talked about. Those are things you should check off in the discussion to say, are we really getting the whole picture of everything we're talking about? And my interest is both animal and human health, so last slide please. And so with that interest, and just having had a son who had a very traumatic leg injury, and everything's fine now, but we went through some times in the hospital where we ended up going to vancomycin to save him and his leg. I still come away from that with a concern that what we do in the name of human health, I'm for everything we can do for human health, but my concern as a veterinarian is I don't want to see something done in the name of human health which may have no or minimal effect or a bad effect on human health and then harms animal health in the process. And that's why I'm here speaking today, is to make sure that you check off those things as we discuss and consider the whole picture in our discussion.